Before drugs make it onto the pharmacy shelves, they have to be tested. New drugs are constantly being developed, but before they can be given to the general public, they have to go through a thorough testing procedure to make sure that they're safe and effective. There are 3 main stages in drug testing: preclinical testing on human cells and tissues, preclinical testing on live animals, and clinical testing on human volunteers. In preclinical testing, drugs are tested on human cells and tissues in a lab. However, you can't use human cells and tissues to test drugs that affect whole or multiple body systems. Like testing a drug for blood pressure must be done on a whole animal because it has an intact circulatory system. The next step in preclinical testing is to test the drug on live animals. This is to test for efficacy, whether the drug works and produces the effect you're looking for, and to find out about its toxicity, how harmful it is, and to find the best dosage, the concentration that should be given, and how often it should be given. The law in Britain states that any new drug must be tested on 2 different live mammals. Some people think it's cruel to test on animals, but others believe this is the safest way to make sure a drug isn't dangerous before it's given to humans. Other people think that animals are so different from humans that testing on animals is pointless. If the drug passes the tests on animals, then it's tested on human volunteers in a clinical trial. First, the drug is tested on healthy volunteers. This is to make sure that it doesn't have any harmful side effects when the body is working normally. At the start of the trial, a very low dose of the drug is given, and this is gradually increased. If the results of the test on healthy volunteers are good, the drugs can be tested on people suffering from the illness. The optimum dose is found. This is the dose of the drug that is the most effective and has few side effects. To test how well the drug works, patients are randomly put into 2 groups. One is given the new drug, and the other is given a placebo. A placebo is a substance that is like the drug being tested but doesn't do anything. This is so the doctor can see the actual difference the drug makes. It allows for the placebo effect, when the patient expects the treatment to work and so feels better, even though the treatment isn't doing anything. An example: a pharmaceutical company has developed a drug, Drug X, to help people with high blood cholesterol to lower their cholesterol levels. The drug was tested on 1,500 patients with high cholesterol. The patients were divided into 2 groups, and over 6 months, each patient had 1 tablet per day. One group had a tablet containing Drug X, and the other group had a tablet without Drug X, the placebo. Clinical trials are blind. The patient in the study doesn't know whether they are getting the drug or the placebo. In fact, they're often double blind. Neither the patient nor the doctor knows who is getting the drug until the results have been gathered. This is so the doctors monitoring their patients and analysing the results aren't subconsciously influenced by their knowledge. The result of drug testing and drug trials aren't published until they've been through peer review. This helps to prevent false claims. It's really important that drugs are tested thoroughly before being used to make sure they're safe. An example of what can happen when drugs are not thoroughly tested is the case of thalidomide. Thalidomide is a drug that was developed in the 1950s. It was intended as a sleeping pill and was tested for that use, but later it was also found to be effective in relieving morning sickness in pregnant women. Unfortunately, thalidomide hadn't been tested as a drug for morning sickness, and so it wasn't known that it could pass through the placenta and affect the fetus, causing abnormal limb development. In some cases, babies were born with no arms or legs at all. About 10,000 babies were affected by thalidomide, and only about half of them survived. The drug was banned and more rigorous testing procedures were introduced. More recently, thalidomide has been used in the treatment of leprosy and other diseases like cancer. That brings us to the end of the B3 module. We've covered all of the sections that we need to be able to pass infection and response. I look forward to seeing you in the next topic.